Hi everyone, welcome to Medical Microbiology. Today we will continue chapter 4, Structure of Bacterial or Prokaryotic Cells. On lecture 3, we will discuss about cell envelope, which is the cell wall and the cell membrane. Let's see the summary of the structure of the prokaryotic cell. Remember, it has three main structure. The first one is what we call the external structure. Okay, so external structure that consists of appendages and glycocalyx. The appendages mostly used for movement like flagella and axial filament, or may be used for attachment like fimbriae, or used for transferring the genetic materials like pili. The glycocalyx, it's used for protection and also to produce the biofilm, which is the attraction for other bacteria to come and to make the community of microorganisms. The second structure, okay, the second group of structure is called the cell envelope. This is the one that we will study today. Okay, the cell envelope of bacteria or prokaryotic cell consists of two main layers. The first one is called the cell wall, and the second one is the cell membrane. Okay, again, the cell envelope. The cell envelope consists of cell wall and the cell membrane. The primary function of the cell envelope is for covering, protecting the inside structure of the cell. It is also used for maintaining the cell integrity. It's also used for movement of materials in and out. There are two different groups of bacteria, okay, based on the cell wall. And this is the two main group of classification of bacteria. The first one is called the gram-positive bacteria. Okay, it has a very thick cell wall. And the second group is called the gram-negative bacteria. It has a thinner cell wall. Okay, so that the two main group of bacteria based on their cell wall. Now the cell wall, the structure again mainly, okay, the main molecules that make up the cell wall is called peptidoglycan. So this is the main molecules that make the cell wall of bacteria. Remember, algae, plants, fungi, they also have the cell wall. But the molecules that make their cell wall will be different compared to the molecule that make the cell walls of bacteria. So the cell wall of bacteria made of peptidoglycan molecule. And from the name, we should be able to guess, okay, that this molecule actually consists of peptide or peptide, which is protein or amino acid, and the glycan, which is glucose or carbohydrate. So this molecule actually composed of amino acid and carbohydrate. Okay, this is it's a lot of amino acid over here. This is all the amino acid, and this is the carbohydrate. So this carbohydrate connected with this peptide or the amino acid. There are two types of bacteria based on the cell wall. The first one is called the gram-positive bacteria. Okay, so gram-positive bacteria, which is, look at this cell wall over here. The cell wall is really, really thick. So the peptidoglycan, okay, the peptidoglycan wall is very, very thick. So this is gram-positive bacteria. And then under that cell wall, of course, this is the cell membrane over here. Okay, so this is cell membrane, and this is cell wall. So that's the gram-positive bacteria. The gram-negative bacteria, if you see the cell wall, is very thin, and this cell wall is actually located between the cell membrane and other protein layer on the top. Okay, so this is the cell wall. This is the cell wall, which is the peptidoglycan over here, which is really, really thin, okay, compared to the gram-positive bacteria. And of course, this is the cell membrane. Okay, cell membrane. And the top, there is another layer of proteins, okay, which is outside, okay, outside the cell wall. So basically, this gram-negative bacteria has 3 layers on their cell envelope. Okay, the protein layer, the outer membrane, okay, and then the cell wall and the cell membrane. Okay, so 3 layers over here. 1, 2, and 3. Now, if we compare side by side, again, you see that this gram-positive bacteria only have 2 layers, okay, of the cell envelope. The first one is this one, which is cell wall. And this second one is this, which is cell membrane. Okay, and this is the gram-negative bacteria. It has 3 layers. One, this is the outer membrane. Outer membrane. This one is the cell wall, which is really thin. And number 3 is the cell membrane. Okay. So these are difference between the cell wall of gram-positive bacteria and gram-negative bacteria. So during the lab experiment, you will do the experiment, which is staining the bacterial cell, and then from the stain procedure, the gram staining, you will be able to know which one is gram-positive and which one is gram-negative. The gram-positive bacteria will have blue or purple color under the microscope. The gram-negative bacteria will have red or pink color under the microscope. This is another comparison in the table. Okay, again, the number of, this is cell wall. Okay, so cell wall, the number of layer of gram-positive only one, which is really thick peptidoglycan. This one has two, okay, which is outer membrane and thin peptidoglycan. Okay, so this one is thick, oh sorry, peptidoglycan. Okay. Again, chemical composition of the cell wall, the main molecule is called the peptidoglycan, and there are some other molecules that also present on the cell wall. For the gram-negative bacteria, okay, of course, peptidoglycan, but it's really thin. And above that, there is a outer membrane that consists of lipoprotein, lipopolysaccharide layer. And then the thickness is really thick, between 20 to 80 nanometer. And gram-negative bacteria is only between 8 to 11 nanometer. Outer membrane, no. Okay, there is no outer membrane. This one, yes, it has the outer membrane, which is consists of lipoprotein and lipopolysaccharide. Periplasmic space is very narrow, and this one is very wide or extensive. Permeable to molecule is more penetrable, and this is less penetrable. Okay, this is also give us the idea that the gram-negative bacteria will be more difficult to destroy by antibiotics. Okay, because they are less penetrable. This one is more penetrable, mean it's gonna be easier to destroy by the antibiotic. Now, the gram stain, which is the staining procedure to identify whether bacteria gram positive or gram negative. Okay, so again, gram positive will stain purple. The gram negative will stain red. Again, you will do this in the lab experiment. So just follow the procedure, the instruction during the lab experiment. And finally, if you do it correctly, you will be able to know or to identify which one is gram-positive bacteria and which one is gram-negative bacteria. Okay, that's the two main type of bacteria based on the cell wall, which is gram-positive and gram-negative bacteria. But there are some exceptions. There are some other bacteria which is actually really not many of them do not have the peptidoglycan. Okay, so they have different type of molecules that make up their cell wall. Okay, for example, is the TB bacteria. Okay, tuberculosis bacteria. They do not have the peptidoglycan. Okay. Their cell wall is actually consists of lipid mycolic acid, okay, which is gonna be difficult to stain with the gram staining. It means that they cannot be stained with the gram staining. You will not be able to tell whether they are positive or negative. Therefore, they have special staining procedure, which is called the acid fast staining. Okay, so the acid fast staining is especially used for this type of bacteria. Like for example, to identify the TB bacteria, then we use the acid-fast staining procedure. You will also do this in the lab experiment. So just follow the instruction, follow the procedure, then you should be able to see whether the bacteria is TB bacteria or other bacteria. And there are also some bacteria that actually do not have the cell wall, and this is very rare. It's only group is called the mycoplasma. Okay, so mycoplasma is a group of bacteria that do not have the cell wall. Okay, no cell wall in this group, and this is the very rare type of bacteria. So mostly bacteria have cell wall except the mycoplasma. Now under the cell wall, okay, there will be a second layer is called the cell membrane. Remember when we talk about the differences or the similarities between prokaryotic cell and eukaryotic cell, all cell, whether this cell is prokaryotic cell or eukaryotic cell, they all will have the cell membrane. Okay, so all cell have cell membrane. Okay, but not all cell have the cell wall. Okay, cell wall only present in bacteria, plant, algae, fungi. But protozoa, animal, they do not have cell wall. But all of them, okay, all of them have cell membrane. So cell membrane is the layer that present in all type of cell. And they're all the same type of molecule that make the cell membrane is called the phospholipid. Okay, it's called the phospholipid, which is the molecule that has phosphate and lipid. Okay, fat. Okay, and they make two layer. Therefore, it's called the bilayer. So this is two layer of phospholipid molecule. If you see this one, this is one phospholipid molecule like this. It has head, okay, and then two tails. This is one phospholipid molecule. So they make layer. So one layer on the top like this. This is one layer, and there is another layer at the bottom. Okay, so it has two layer. This is what we call the bilayer. Okay, two layer of phospholipid molecule. So you see this one. This is one layer. Okay, and this is another layer of phospholipid molecules. So therefore, the cell membrane is also known as a phospholipid bilayer because it has two layer of phospholipid molecule. In addition of this main structure, which is phospholipid molecules, the cell membrane also have other molecule like proteins. So this one, this is a protein. This is another proteins. This is proteins. This is other proteins. This is some carbohydrates over here. Okay, so and it is okay. This one over here is cholesterol. Okay, so the main structure, the main framework is phospholipid bilayer, but there are some other molecule like proteins, carbohydrate, and also cholesterol present in the cell membrane. And the primary function of the cell membrane, of course, for protection and also for controlling movement of molecule, okay, from inside to outside or from outside to inside. Okay, I think that's enough for the cell envelope of bacterial cell, which is the cell wall and the cell membrane. Just remember, the cell wall of bacteria consists of peptidoglycan molecules. The cell membrane consists of phospholipid bilayer. We will see you again on the next lecture, which is internal structure of prokaryotic cell. Goodbye now.